And I will share my screen. All right, so slideshow. We will play from start. Okay, so prior to everyone coming on, we were playing Mercy, Mercy Me by, why do I keep forgetting his name? Ellis, what's his name again?
you got me on that one. Let's see.
Marvin Gaye. Marvin
Gaye.
Oh yeah, Marvin Gaye.
How could I forget? Our Detroit boy. Yes.
That is this song that got our group a grant. We used
for
it
real?
in a video interview. Yes. I'm
Wow.
offended. Marvin Gaye.
I know. He's like one of the favorites all time.
All time. Yeah.
I forgive you.
Thanks. So anyway, that's our theme song today here. Again, I want to emphasize that the picture that you're looking at, bring to your attention that it's considered to be the most iconic photograph in human history. It's called Earthrise, and the QR code there will take you to the interview of the Apollo 8 astronaut, Bill Anders, who took this picture. It's a picture of our home. It inspired Earth Day. It inspired a lot of environmental movement. And so paying tribute to that photo and to what came from it. And as this slide here shows, the United Nations Environmental Program has designated as Earth's, our home's triple threat: pollution, climate change, and loss of biodiversity. I believe today our speaker is going to be in the area of biodiversity. And we'll see, maybe it could be also related to pollution. I'm not sure. We wait to hear what she has to say. But this is part of why we do Environmental Fridays each week, is we try to address these topics. And our goal is to bring awareness, but not just that, we want to bring and encourage action, not just us knowing more, but also doing more. To bring education and to foster engagement. So a couple of things. Again, welcome to Environmental Fridays. This is our 7th season, and you could find us online. This season, we have 15 episodes, 16 guest speakers, and you could see where they come from here. And we have like a photo gallery of our speakers to see all the smiling faces, committed people to the environment there. In October, which begins next week, we'll have 4 presentations. As you could see here, Birds of Tobago. So we'll be going to my dad's birthplace, which is Tobago. Global warming and coral reef bleaching will be considered the next week after that, from Derek, who is with the National Oceanic and Atmospheric Administration in Florida. Then we will look at Future Farm, a really good new technology that's being developed by Michael in conjunction with Art Jones out in New York. Then back to Trinidad, my birthplace, and we will actually have a presentation from Chief of the First Peoples of the Caribbean, and we learn some more about them. We have essentially 2 more spots if anybody's interested in being a co-host with me. We have 2 more spots coming up, November 29th and December 6th. Just reach out to me, murrayD@andrews.edu. It's very simple function. So at this point, I will introduce Tahara. I don't think she's on yet, but Tahara is being, as far as we know, still affected by the storm, by the hurricane, Hurricane Helene, I believe. It's hit landfall, and she's in South Carolina, so I'm assuming that it's making havoc in South Carolina. But in her absence, we will have Juan Ramos, I believe, yep, who will give us a introduction to our speaker today. Both Juan and Tahara are students of our speaker, Dr. Ozbay, and believe know each other well. So Juan, this is your time to introduce our guest speaker for today.
Okay, good morning, everyone. As was just mentioned here by Dr. Murray, my name is Juan. I'm a graduate student at Delaware State University, and I'll be introducing our speaker today, who is my advisor, Dr. Gulnihal Ozbay. She is the Associate Dean for Extension and Research, Professor of Natural Resources, and the Director of the Integrated PhD Program in the Agriculture, Food, and Environmental Science in the College of Agriculture, Science, Technology at Delaware State University. She received her PhD in the fisheries and applied aquaculture at Auburn University, Auburn, Alabama in 2002. She conducted research assisting in the development of aquaculture influent water quality standards in from the U.S. Environmental Protection Agency and her PhD project. She also received her master's in bioresource engineering with a concentration in the marine bioresources at the University of Maine in 1996. And then she also received her bachelor's in the fisheries and aquaculture engineering in Turkey in 1991. She also received her second master's at the time in food science and biotechnology graduate program from Delaware State University.
All right. Thank you so much. And we will now turn our attention. Let me stop sharing. And Dr. Ozbay, you could now share your screen with us. Right. Try to get it into full. There we go. It's all good. Oh, okay. Try again. So it's not in full presentation mode yet. There you go. It's good now. So it's your turn.
Hello, can you hear me okay now?
We are hearing you now.
Okay, great. Good morning, everyone. And I like to thank you, Dr. Murray, before I start my presentation for such a wonderful program. I am so happy to be part of this program. And also, I like to thank you, my students, especially Tahira and Juan, coordinating to introduce me today, and also the whole team. Um, so I am really excited that I like to give you some information and share my journey with my wonderful students who have been very passionate and very dedicated to environmental research and environmental education and outreach. So this will be our story today. And I am trying to move. Okay, so as you notice from these pictures, everything I do pretty much in my 22 years in Delaware State University, even prior to my tenure as a faculty, I have always worked in a team with the team of students and also the volunteers. So, and we have had so much learning experience and fun together. You could see from this picture, students were learning and also enjoying all these natural wonders of Belize. So we are really looking forward to go to Trinidad and other countries in the future.
All right.
And exactly, yes. And here you could see we also have a lot of fun in Delaware Mid-Atlantic area. We are surrounded by water. My field of research require water. I am an aquatic person. And students who does wildlife, who does microbiology, everything, it needs to concentrate on more aquatic, settings. But I like to bring your attention to how many opportunities that we have where the students and the volunteers and the high school students, even the K-12, the much younger students have opportunity to join us with the field trips, with the courses, and we also had opportunity to go to Ghana. So I took the students to Ghana together with the UAPB, University of Arkansas Pine Bluff faculty member. So they actually had a workshop with us. They were the trainer and they did teach the community and the students, faculty there about how to do aquaculture, do the fish culturing. So we have had a lot of opportunities. So as my title states, for today's talk, we are talking about restoration and also biodiversity. And you could and so the basically we are going to start out with those definitions, what those mean. So what are the definitions of them? What are the benefit of oysters? Because a lot of research focuses on oysters. And then I like to highlight some of our student research that directly contributing to these areas. And we will go through, there are all these names. You see, these are all the students, graduate students in my lab. And then we will go through the conclusions, recommendation, and overall purpose of this presentation. So I am not going to read the whole thing, but I wanted to make sure that my presentation is shared, you got the whole picture of why it is important. Habitat restoration is really dealing with damaged or like degraded or distracted habitat. It means that the whole goal of ecological restoration is to return the degraded or damaged system to its historical trajectory. We are not talking about condition. We even are talking about making it better. Uh, so basically, ecological restoration seeks to initiate or accelerate ecosystem recovery from the damage, degradation, or destruction. You could see in this picture that we start, like in India, they start out with the plant, they put the plants in there. Over the timeline of 30 years, they have seen the very, very strong growth of the mangroves. Biodiversity, on the other hand, is referred to variety of living things in any given environment. So we are talking about plants, animal, bacteria, fungi. So you could see this, you can read the rest of the details if this presentation is shared with you. So why it is so important that we work with oysters? Oysters are basically habitat makers. So there are a lot of details that I want to go through very quickly, actually, since we have a limited time. So we are dealing with Eastern oyster. It's a Crossostria virginica. They are really very strong filter feeders. We don't have to feed them primarily in nature because they constantly filter the water. For like any given day, they have 50 gallons um, of water they can filter. They are basically in the brackish to marine habitat. It means require salinity. So generally, we don't want to go down less than 10 And then, up to 30 part per thousand, it means a gram per liter. So when you say part per million, it's milligram per liter. When we say part per thousand, it means gram per liter. So we do look at a lot of environmental parameters, how the oysters interact with the other organisms and how they create habitats. And also we do have animals that eat on oysters. We also pay attention to those. They are really keystone species, as I mentioned about it. They really do feeding. They are filter feeders. They create a habitat for fish and crustaceans. They basically enhance water quality by filtering. It's like a think about what is it called, the filters that we use in the house. Similar concept. They constantly filter. And they really, as they actually pick up all the algae, bacteria, everything from the water, they remove a lot of excess nutrients that way because they are part of the living things, especially algae. So algae grow a lot when you have a lot of excess nitrogen and phosphate. When you have oyster filtering them, they are clearing the water. It's basically shoreline protection and stabilization because they create 3D structure. And a lot of times when you see oyster reefs, they actually create a habitat, very solid habitat, because oyster have to attach themselves to something. So they attach on each other. They create like on top of each other a big habitat. And reducing water velocity this way, and they also enhance sedimentation. They capture it. So that really helps the sedimentation actually being suspended in the water and or moving around. Of course, they increase diversity. The most importantly, species diversity, their abundance, their richness. Oyster aquaculture is implemented in Delaware in 2017 because with the hope that we increase oyster population. And what happened is that many oyster population wiped out because of the parasitic diseases, MSX and dermal, and over 90% of oyster populations gone. And since 1950s up to now, so we are trying to do restoration efforts, and also a lot of people love eating oyster because they are rich in omega-3 fatty acids, and they also provides, like they have all these important iod, the what we call is the nutrients that good for the body. So, but we have to watch out because they are filter feeders. They can accumulate microbial contaminants. There are certain times of the year oysters are safe to eat, but we have to be really cautious. So oyster aquaculture is implemented in Delaware in 2017. We were the last state on the East Coast joined the whole aquaculture efforts. Um, as we as we go through this presentation, you will see they are like ecosystem engineers. They create habitat, they increase diversity. Oh my God, they got so many other benefits that I just mentioned. So they basically, even aquaculture itself, because we put the oysters in a structure, they actually create habitat itself. So oyster in them, because of the inflation, they uh, filter the water. So there's all the other, and you will see from the presentation, there are some increase in the species diversity and so on. Again, I mentioned about this, the due to the decline in oyster populations, we have to restore oyster habitat. So we are also restoring habitat. We are doing habitat restoration. Increasing oyster population leads into habitat restoration for us. So due to also overharvesting, people prior to us, they didn't know. So a lot of the, fisheries are over basically harvested. So degradation of water quality, and when you have water pollution too much in the water, sometimes you can't grow the baby oysters, so they die. And eutrophication is a term here. I want to mention eutrophication means excess nutrients like phosphate and nitrogen. You could see in this system, if you do not have a good, healthy ecosystem, you will see all these pollutants, increased overall aquatic pollutions. And oyster play important role. So in order to get the oyster grow, you still need some quality of water to grow them. So water quality is really important indicator of environmental health. And we actually, according to EPA, Delaware water, especially in inland bays, are considered poor water quality conditions. We need to change that. High levels of nitrogen and phosphates are always recorded throughout Delaware, which means that we are getting a lot of excess, like algae growth and so on. And habitat degradation affects many important species and decrease biodiversity. So we also have to look at that we have a big issues with the seagrass overall area of seagrasses. Seagrass is like equally important ecosystem in in the whole aquatic environment. They provide habitat. So both seagrasses and bivalves mollusk form habitats. When I say bivalve mollusk, oysters is part of them, okay? Oysters, mussels, clams, they are bivalve mollusk. So with the seagrass and mollusk, they form really beautiful ecosystem environmental condition. Seagrass can facilitate oyster calcification by controlling pH levels and mitigate the effects of ocean acidification. And remember, seagrasses does photosynthesis. Do you remember photosynthesis from your class? The plants take up CO2, carbon dioxide. They produce oxygen to the environment, right? Under the sunlight and nutrients and water. So this is a very important process for overall living on the earth system. Bivalves can enhance seagrass size and growth directly fertilizing them, because when oyster filter anything that they do not, it's not algae, they spit it out, or like we call the pseudofeces, and that is a little bit dense, so it actually sinks. So they created like a very rich benthic habitat. So, and as oyster filter the water, they clear it and it makes it light to penetrate better and increases seagrass population. That's what we are observing. Okay, so the global decline overally in oyster population sparked a lot of interest throughout United States and different parts of the world. I don't know if you guys heard about 1 billion oyster project in New York. I was one of their guest speaker, and we talk about like a lot of those restoration activities. In Delaware, Center for Inland Bays is our lead agency. They actually lead this whole oyster restoration effort we call the pilot oyster reefs in Delaware Inland Bays, which consist of 3 bays: Rehoboth Bay, Indian River, and Little Assawoman Bay. But today in my talk, I will concentrate on more on Rehoboth Bay, a little bit on the Indian River Bay. So basically the Center for Inland Bay is a nonprofit. They actually work with the school system like us with the schools, and then also the volunteers, community volunteers. We basically volunteer to help increase the oyster reefs. We also have an ongoing aquaculture leasing in that area. We know that oyster contribute to the environment. Oyster aquaculture does as well, so that we are encouraging people to do those kind of activities in our water in Delaware Inland Bays. But not everywhere is suitable. Some areas are habitat for birds, others so they are all determined by the state agency like DNRAC. They do have a map where you can put your oyster aquaculture activities or the reefs. So oyster farming is done through the shellfish leasing, and that has to be approved by DNRAC. DNRAC means is Department of Natural Resources and Environmental Control in Delaware. So I am just going to go over all these student research very quickly. The first project, one of the first project that we started, funded by EPSCoR, we focused on biodiversity. We wanted to know, like, how the aquatic diversity increases, their abundance, diversity increases, how the basically those pilot oyster reefs shells. When you do pilot oyster reef, you have to introduce substrate like oyster shells, clean oyster shells. So how, and then also oyster aquaculture, all the cages and everything that we put out there, how they increase biodiversity in that area. We are, in order for us to see the association between environmental stress and how everything is going in the whole area, how all the dynamics works and interactions, we gotta monitor water quality, collect and all the species that we actually see around the oyster reefs, and then we also look at some of the predatory species. They actually you know um rely on oysters for food, and we monitor the oyster recruitment. When I say recruitment, baby oyster has to set. In order for to survive after 4 weeks, they need to be able to set on hard substrate like a shells, or hard substrate like a reef that is constructed, artificial reefs and such. And these are the study site. It's a Delaware Inland base. And again, it is the Rehoboth Bay. We have 4 study sites on the west side of the Rehoboth Bay. And these are labeled. So we have 3 characteristics in our site. West side of the bay is close to ocean site. And west side, which is with the 4 sites on the map, you see, is that actually these are, as is labeled here, these are places where there we have oyster aquaculture activity on the left side, east side. We have oyster reefs, like a restoration activity on the east and left side, and the control. Again, when you do any experiment, you have to be able to compare it with something. If we consider oyster aquaculture and restoration like a treatment, right? Because we are introducing oyster in that area, but we have to compare those, or data be collected in those areas with something that there's no oysters. That's why we do control. So you could see from these maps. And this is in Delaware, Rehoboth Bay, in Delaware Inland Base, but Rehoboth Bay. So you have to learn how to use some of the instrumentation. A lot of research is done through the equipmentations, sensors. So these are some of them that we use YSI. So the on the left side, this instrument on the left is the multi sensors, and we use it in the field, and we monitor temperature, pH, dissolved oxygen, turbidity, and all the other parameters. On the right side, it is for the lab. So you bring in the water samples and monitor nutrients like all the alkalinity, hardness. Nutrients, we are talking about nitrate, phosphate, and so on. And so how do we do species diversity monitoring? So species that, again, we need a hard substrate. So this particular project, we use the clean shells, introduce them to the environment. There are certain designated area we have, and then we actually monitor them. They are actually, those shells were in the bags, and they were put, like bunch of them put in there. How did we do diversity? We basically introduce those like a shell bags to see how many organisms are attracted over time. So, and as you notice, we probably left them several weeks out there. And then we bring them in, and then we spread them, and we start collecting specimens, and also writing down how many organisms we recorded it, how many kinds of them, and so on. And I'm not going to get into this as like in detail, but important part in this message is that every time you do research, you got to apply statistic to it. And you could see from our charts, we have all kinds of organisms. We have reported it. We had abundance of mud crabs in those, like a lot of those study sites, even including control, but many of our diversity is recorded where the reefs are built, and then followed by the aquaculture site. And you could see from this graph 5 most common species that we actually identify as mud crab, worms, amphipods, toadfish, oyster drills. Oyster drill is a predatory to oysters. They actually kill them. And then again, if you are doing research, you need to have this some understanding of statistic. You need to have some understanding of how you actually use Excel. Take the averages. So these are the box graphs, we call it. It's run through the statistical software, and it shows you the mean values, what you are analyzing on mean values, and help you understand how wide your standard deviation is. Standard deviations are basically how close your research data to each other. Is there a big differences? And so you could see some of the boxes are big, but in general, we see the same trend. The oyster reef increases diversity and species abundance, as well as species richness, and again, abundance, diversity, and richness altogether. And oyster recruitment also is higher in general where the oyster reefs are. Second most highest, higher recruitment is aquaculture. So this is like a trend. Oyster reef increases the most, and then oyster aquaculture second, and so on. And then control, there is no oyster, you don't see anything in some of the control sites. We do this over time. I forgot to mention, we selected warm months. We didn't do it throughout the year. It's very hard to be there in the wintertime on the boat and be in the water. So we prevented. What we did is until it gets really cold in November, we did our research from June through November. And sometimes we start in May, but we don't use the May data because we are still adjusting all the sites. And so we collect about 5 to 6 months data. So the presence of structural habitat provided a good, conditions for oyster baby to set. So these are the diversity result summary. Species, again, richness, abundance, and all the diversity is much higher. And then we look at the overall ecological benefit. We see that reef provides the most protection and better habitat. So, and then Tahira herself also look at the biodiversity, but she's a wildlife major, and wildlife people, my major people use a lot of, camera traps, and she has this interest, strong interest. And I told her, let's do sub submerge, or like a underwater camera traps, and to see that if we can have a good look at some of these species. So, but she also did, since she has a very strong genetic backgrounds from her undergraduate degree, we also integrate environmental DNA. So she actually looked at environmental DNA signatures to confirm her data. So basically, she gives the detail in this presentation, like what she did, how long she put the cameras, and how long she waited, and like every 2 3 hours, and then she monitor like whichever organisms is going in and out that area. In her research, she concentrated on 5 study sites instead of 7, because we didn't want to have, there were some study sites in our previous research with memories research. We didn't see a lot of activities. We concentrate on the ones that we could see a good recording. And also water clarity wasn't very good in the other 2 sites. We couldn't see anything. And environmental DNA is a new concept for us, really. And thanks to Dr. Ali Parsamir. So he is a research scientist in my lab, in One Health Lab, and he has introduced a lot of those concepts to this, research with his expertise in this area. And basically we collected water and soil in those study sites, and then she basically looked at some of the DNA signature. This has a lot of processing. So if you ever involved with any genetic and genomic research, you have to be, in any other research actually, you got to learn to be patient. It requires steps. Some people are very good working with smaller volumes, because with eDNA and any other RNA, you got to really work in a sterile, very careful conditions under the hoods, and no contamination risk. and you got to be really careful. You're working with very, very smaller volumes. So it requires a lot of steps. You got to do all these steps that is here to isolate the eDNA from your samples, from water and the soil. And then you also need to learn how to run real-time PCR, polymerase chain reaction. It means you are multiplying the DNA to be able to have a better understanding if the species is present in your samples. Sometimes you use eDNA also to find overall quantity. So, and you could see in here, it requires some steps again. And here you go. We got the results. And there were, so according to her, assessment, all the recordings, she got about 23 species. She is trying to confirm them, and this is a very good results. And she actually get a lot better, connections. So you, and basically, with between eDNA and camera trap, she was identify species that predatory to oysters. She also identifies species that maybe we didn't see in the previous research. So it was, we have a great number of species and individuals observed in the reef system. Greater, and this confirms the study that we did with memory earlier that I described. Greatest number of individuals was like Atlantic menhaden and silverside. And there are all other species that I will show you. In the camera trap, you could see horseshoe crab on the left, right? And then you have summer flounder and black sea bass, and you even have turtle in here, and you even have the bird, the cormorant. So you could see with all these, like cameras. And eDNA also confirmed, right? So we do have horseshoe crabs and some predatory organisms. But one problem with eDNA, you need to have what we call is the um, primers. When you use the primers, it means that the species of interest or whatever target gene sequence that you want to use, you need to have a forward and backwards, the primers. You need to be a little bit specific. So instead of doing entire species, we concentrate on the ones that are more threatening to oysters. So, and she also did other studies. I'm not going to get into very, very detail on this, but what happened is that she used water quality parameters like pH to see if the oysters are having hard time. They are calcifying organism. They build their shell. They need calcium, right? So they basically have to build their shells and grow. We look at some of the we call aragonite and calcite values. These are the chemical conditions that must exist for, especially in marine environment, for calcareous organisms, like also reefs. Remember, coral reefs. So these things are very important. If we are threatened by ocean acidification, it means they are going to suffer. So, and in order to do this study, they all needed to, this is actually done to get primarily by high school student help. He was a data science student. He was amazing. And then Tahira literally worked with him, integrated what he knew and into her research as a graduate student. So they actually run the software and they actually use the water quality parameters that we collected to understand the overall conditions. And you could see they use the pH values, alkalinity, calcium hardness. So pH was very consistent throughout the years for us. Alkalinity fluctuated, so it gives you some alarming thing. Calcium hardness also fluctuated. So depending on the season and the time of the year, it changed. But using these parameters, we are able to identify aragonite and calcite values. So basically, we didn't directly monitor aragonite and calcite. We put them in a software, like we put pH, alkalinity, calcium, temperature values in a software package to understand the parameters for aragonite and calcite. As you notice, we consider one is our, we want to have one is basically is our limit. Anything below 1 is we call it start, organisms are under the stress. So it means that pH is start getting lower. Ocean, there is as you noticed with ocean acidification, that will even get lower and lower. So we want to make sure calcifying organisms are not suffering. And you could see they also, this is actually done with the help of Abigail Armani and Kelvin Ofori, the graduate students in our human ecology department, and they actually run this for their studies, and Tahira was able to use for her own studies, looking at associations and correlations between different water quality parameters. Lighter colors indicate strong positive correlation, while the darker ones doesn't show any correlations. And based on these parameters, we were able to identify what water parameters are very strongly associated. Again, this is called the heat maps, and we also monitor it. We are looking at for different years. We did this study, all these projects, either 2 years or 4 years timeline, but all the water quality is 4 years timeline. And at the end, artificial reefs had more biodiversity, and it is confirmed through the real-time monitoring with the cameras. eDNA really help us to confirm some of the predatory organisms. Um, so physical water quality parameters for the most part met, but still we got a little bit stress in the environment in some of the months that we have to pay attention. Chemical parameters really fluctuated. And so aragonite and calcium saturation states were undersaturated during certain months, especially warmer months. And so Mohana did, like, literally her research is focusing on where. So basically all around the Delaware Inlet Basin, we have housing complex, and we have a wastewater treatment facility, we have a poultry farm, all the other activities, the boat ramps. So with the exception of point pollutants, we call point pollutants comes from wastewater system, and at some extent industry, all the others like lawns and boat ramps, the agriculture, they are all considered non-point sources. So when we have a non-point sources, it is very hard to track where the nutrients are coming. Basically, in her study, she used the chemical signature to identify associations of nutrients chemistry in the water, in the sediment, with surrounding habitat. And we are talking about soils. So she had to use water quality parameter as well as all the, we call the isotope. It means that it helps you track where origin of the nitrogen. So basically, the whole idea for us in this project is to move on to identify this non-point, to make it more like a point system, like a points where to point them where they are coming from. And you could see here in ammonia nitrate orthophosphate. And then she also did additional work, because since we study with oyster, she looked at if the oysters are stressed. So she looked at their growing condition, the condition index. And then we did a lot of interactions between certain water quality parameters in her study. So all these projects, and here goes Juan Ramos. He did it a little bit different than others. He has a very strong interest working with crustaceans. I talk about bivalve mollusk earlier. So blue crabs are crustaceans, and they are delicacy for overall in the East Coast. But at the same time, they are also very important keystone species. So basically, blue crab populations start declining in the mid-Atlantic area, start alarming people. And first recorded by IMET, we call the University of Maryland Marine Center. They recorded some threatening virus, reovirus, Calinectectus staphytis, which is the scientific name. And in Rehoboth Bay, we did not have any recording, monitoring. So basically, Juan's research focused on if this potential threat exists in our study sites. And he also looked at the overall population characteristics of blue crabs in that area where these study sites that I show you, where the oyster activities. This has to be done in relation to the study sites that we identify. And he deployed 3 cages per site. So he had 6 study sites. And then he basically monitor blue crab width, length, sex, and all the other parameters. And he also stores some of them for, again, genetic work. And with the help of Dr. Eric Schott and as well as Dr. Ali Parsamir, he was able to do some genetic work to identify the presence or absence of these threats. And so by looking at the overall characteristics, so he identified that so first of all, we monitor some of these we call the virus, and 2.3% detection out of 88 individual crab. So still we hope that we didn't see it, but we have it. And then you could see that we are able to identify how many crabs are, egg bearing crabs, females, males, and water quality in general is very good for blue crabs here in Delaware Inland Bays. So there he recorded more juvenile blue crabs where the oysters are, so both restoration site and aquaculture site. And Calvin, on the other hand, he's a part of food science and technology program students, like a graduate program. He wanted to focus on, okay, so we got all these monitoring going on in terms of oyster conditions, oyster health, ecological. How about people consuming these oysters from aquaculture side? So he wanted to do a work on more microbial contamination risk. Like some of these bacteria that when you consume oyster raw ends up, like when you consume it, the higher concentration leads like diarrhea, nausea, different side effects. So basically he wanted to concentrate on that, and he wanted to look at this in relation to water quality. So it is very, very important to understand that anytime we do consume anything raw, there is a risk associated with microbial contaminants. So, and he did a lot of work, microbiological work in the lab. Although, as you noticed, he also went out with the team and collected his oysters water samples. And so, and then he did work with the regular microbiological process. And then he also ran for the genetics. Genetic results demonstrate the presence of bacteria and pathogen in oyster and water samples, especially Vibrio parahemolyticus. So we had abundance of that. And here goes the very last example I want to give. Emily was interested in bacteria that kills the oysters. So again, Emily and Calvin use aquaculture study site. And again, both of the students have high school and undergraduate interns working with them involved in these projects. So for Emily, she concentrated on like a Vibrio tubiaceae or Vibrio paralyticus. These are the ones that doesn't kill us as a human, but it is damaging and killing the baby oysters and affecting overall oyster abundance. So she also had to monitor water, look at the oyster conditions, are they growing? And then the Vibrio species at the aquaculture site. And I like to mention this. We had a wonderful partner for all these projects, especially the aquaculture study sites that we did extensive studies with bacteria, and Mr. Casey, Mark Casey. He's the president of the Delaware Aquaculture Association. So she looked at, all these parameters for both oysters and seawater, and Vibrio detected in oyster sample, and she monitored that the condition index was pretty much similar across the seasons for both years. So with exception of Calvin, all the students had a monitoring for at least 2 years. So overall conclusions, how am I doing, Dr. Murray? Like, I just have a few more slides. Am I doing okay?
So give
Okay,
us
so
maybe about 5 more minutes.
okay, very good. So overall conclusions, oyster restoration is really possible. It means that, we do have a still good quality water in the system to basically grow or restoration activities. So a restoration effort increase wild oyster population because we have a recruitment. When I say recruitment, in the wild, baby oysters growing. And we have the structured habitat because the oysters are creating 3D structure, especially at the reefs. We do have increased abundance of biodiversity and or their abundance and richness. And when I talk about their richness, imagine instead of having like one horseshoe crab, you have multiple horseshoe crab. And so you might have like, let's say you got 10 different species. And for 10 different species, one species have only one animal in that site, but the others have like 100 or third one, another one has only two. Richness, we want to look at the value. It means the balance, overall balance. So instead of having just one species having one, we look at it and say, oh, 5 of these species and 6 of that, so there is some balance in the system. So it is clear that oysters are really strong ecosystem engineers, and it is very, very strong, indicator for overall environmental health for us, in overall the estuary health. Oyster aquaculture provides important role with nutrient removals. So one thing that we haven't tapped into, which this will be the next project for us, is to look at the efficiency of oyster for the removal of nutrients. As I mentioned earlier in my talk, they are filter feeders. They actually filter the water. They even get all the little detritus and everything else, algae. As they filter, we are talking about a microscopic level. And as they filter this, they remove a lot of nutrients that is part of the living things. It's part of the living things. The phosphate, we are part of like nitrogen and phosphate here. We have like carbon, nitrogen, phosphate. So any living thing have those components. So and excess nutrients increases algae. Oyster filter those algae. And then aquaculture, when you remove oysters from the system and people eat, they build on their flesh and people eat. So we basically remove those nutrients through aquaculture. So anyway, the ecosystem stability, we saw that aquaculture site also provided that diversity and provides habitat for many species and also submerged aquatic vegetation. We start seeing increased clarity in oyster. This increases light penetration. In order to do photosynthesis requires sunlight so that we can get the submerged aquatic vegetation to grow. And so we need to continue monitoring. What are the recommendations? We need to continue partnership. You can't do this alone. All these projects, all of them, have, from myself as directing them, Dr. Ali, technician, graduate student is actually selecting this project as their primary. And the other, fellow graduate students, they have undergraduate interns. We got high school students and sometimes volunteers like farmers helping students with these projects. This was like a whole team. And we got also partners with students, experts serve in the students' committees. And the experts help them how to use statistic, and experts help them how to do genetics work, like Dr. Todd. Dr. Todd is basically assistant professor at DSU, and then she basically opened her lab. She does genomics work, opened her lab for our students, and all my lab actually end up doing all the genetic work in her lab. So we need to encourage community to involve, take part in restoration activities. We have to keep monitoring. We have to keep monitoring to show the overall positive impacts of introducing oysters, oyster reefs, and also conducting aquaculture operation to improve the overall restoration that we envision for Delaware Inland Basin, improve water conditions. So we are not coded by EPA under the poor water conditions for Delaware. And love science and have fun doing the outdoor activities. What is the take-home messages? This is literally my last slides for talk. So we had to use very integrative approach in our research, right? Because we had to think about from every perspective. We wouldn't be able to conduct a research with these complex problems. We are talking about environment. Oh my God, so many variables. Without proper training or background, also partnership, we wouldn't be able to do this. In this presentation, we focus on one of the Delaware Inland Basin system, which is the Rehoboth Bay. We would not be able to conduct our research if we didn't have the background and training in the following areas. So if you didn't understand ecology, environmental science, you wouldn't know how the species system works. You know, why those are the aquatic organisms. How do they breed? And where do they exist? And what do they need? Water quality. We had a lot of water quality monitoring in this study. You need to know the chemistry really strong. And so, and oyster physiology. And we are talking about microbiology because we have a students is doing a microbial work. We look at the bacteria affects oysters, you know, is threatening to oyster itself. We also look at the bacteria is threatening for human consumption, you know, for human, because I wouldn't say exactly some of them are not threatening, but it is still like diarrhea and the other, you know, secondary cause could cause a lot of stress to people. And when we look at these parameters, you need to have an understanding. We are talking about environmental chemistry, talking about nutrients. And you one more thing I forgot to put in here is the sensor. You need to have a little understanding about sensor how to use instrumentation. And then as you notice, like in the first student research that I feature in this presentation, memory, and so I mentioned about statistic and math. How are you going to be able to understand what numbers means? Like, what is the statistic? We have to run all our data through statistic to see to get a meaningful outcome. Okay, how are they closely related? Okay, so which site has the high concentration? You look and how like scattered our data is. And all these parameters. We these applicates over the months, we got a lot of data collected. You need to have the meaningful outcome for this. Data science, we did aragonite and calcite. So we didn't monitor aragonite and calcite valleys, which again are the chemistry, water chemistry that helps oyster to grow their shells, the calcareous, and also all the others like blue crabs. Blue crabs also grow their shells, oyster reefs. So you could see all these software skills. You need to be able to understand writing and reading, everything. When you do involve in a project like this, you are developing those skill sets. And there are so many people to thank for. The students that I didn't highlight their projects are listed in here. And again, very special thanks to Dr. Ali. He is like a really important component of my lab, One Health Lab. And the students you could see here, I didn't mention some of the student project because they didn't work directly in Delaware Inland Basin, but again, all these undergraduate students at the bottom right, You could see undergraduate students, volunteers, faculty, scientists, high school students together with us. And so we did a lot of presentation. And what do? We bring science to people. We work with cooperative extension and tell them about our stories. So that all the younger kids learn about what we do. Not only the high school students, we also bring it to elementary school and the middle school. Thank you. Thank you for your attention.
All right, thank you very much. Thank you very much. So I think everybody on this call has just experienced somebody who loves what they do. She was
Oh,
very,
wonderful.
yes, you definitely had very passionate, it was information rich. And so I want to thank you very much for this presentation. Before I ask questions and invite the audience to ask questions, I want to recognize that with us today is Deborah Shaw. She is the EPA administrator for Region 5. And I didn't know she was interested in oysters. But Deborah, if you are there still and want to say a few words, we could have you say something to us.
Professor Murray and Professor Ozbay, thank you. I'm interested in habitat restoration. That's my volunteer activity for 30 years. But I'm also interested in learning whether there is any freshwater counterpart to the oysters that could be deployed to address our algal blooms in our lakes and streams here in the Midwest. But I'll be contacting you, Professor Ozbay, about that.
All right,
Oh,
that's
we
really
look
cool.
forward to it. Thank you.
Yes,
Yes, I look forward to it.
yeah, we were surprised. I was surprised. Oh, Deborah Shaw, she did give us a presentation, I think maybe a year, 2 years ago. So it's good to have her back. So anybody has questions that or comments? I have a lot, but I'll have people, get their questions and comments in.
I'm just wondering, since oysters remove so many things from the water, whether they are a possible consideration for part of the environmental cleanup of water.
Can you repeat your last couple, your question? I miss your last couple of words.
Whether oysters could be used to help with environmental cleanup of water, since they remove so many things from the water.
Oh, yes, absolutely. Oh my God. They are very, very efficient filter feeders. Oh my God. Yes. And so I wouldn't even hesitate. One of the biggest problem with oysters, they are delicacy and edible. So if you use them to clean up the water many times, for instance, wastewater treatment, right? You can use them with the wastewater treatment, as long as there are areas where for after the primary filtering, they could actually filter a lot of nutrients from the water, and it could even they pick up bacteria, detritus, like in different ways that they clean. They don't necessarily eat all of them, it doesn't go through their digestive system, but what they do, oysters does, is that they have a feces and pseudofeces. Pseudofeces is where they pick up the clays, suspended materials that they don't like it. They actually put it in a mucus bond and they spit it. When they spit it, it's heavier. The molecular weight is heavier. It actually sinks. They actually enrich the worm population. I didn't even mention about our very, very previous studies, how much they enrich the benthic community. They actually increase the worm population in the, polychaetes, and it brings a lot of species in that area. But oyster is the very, very effective filter feeders. But anybody using oyster for cleaning the water, they need to be very careful. People don't listen, they don't look at carefully. They try to pick up those oysters for consuming. They might end up getting sick because you are using them as a cleaning the water filter feeder, and when they actually eat them, it causes problems, especially warmer months.
Mm.
Warmer months, we have to be very careful.
So I think Director Shaw also wanted to know about oysters inland, like in the Midwest with the lakes and stuff. I think something that would be of interest here is you spoke mostly of oysters in an ocean setting. What about oysters in lakes and rivers and, inland?
Okay, so generally we have mussels. For instance, in the freshwater settings, so oyster cannot really survive very well. I actually did a study with my undergraduate students and who actually used the freshwater input for in Jamaica. We actually did the Jamaican research in Jamaica, and she looked at the oyster growth conditions when the high river flow in certain areas. And so we went down as low as like 4 part per thousand, which is like actually one condition was almost down to 2 part per thousand, which is like a gram per liter of salinity. And oyster really stressed so bad. I think we lost those oysters. And so I remember oysters has a requirement for salinity or tolerance for salinity, but we have a number of species. I tell you this, I don't know much about Michigan freshwater, mussels, and like I do know some few species, but I am not very familiar. But I tell you this, we do also study in the freshwater system with Elliptio complanata, which is one of the species that we see more in Delaware rivers and waters. And they do not require a hard substrate like oysters. They actually need a very delicate environment. They have a water depth issue. If they are the water depth is very deep, the light doesn't penetrate, and if the nutrient is too high, it's like some of these mussels are very picky, and they need the host fish, right? So different than oysters. So oysters have babies released they fertilize it and they release to the water. And those babies like start like going all over the place. They are mobile and going all over the place in several weeks. They need to settle on a hard substrates. But for the mussels, they need fish to basically carry on their eggs, and they need the fish to actually spread their offspring in the water column, like a gobies,
Okay.
shiners and gobies, and yeah, that's what they need. So they have a little bit more complex relationship in the freshwater system
Okay.
with them, and mainly like mussels, clams, and so zebra mussel, for instance, grow on all different kind of conditions. They are very opportunistic. We even found them in a freshwater system.
Oh, okay.
But we also found a lot in the wastewater facility discharge area where the estuary meets. Yeah.
Okay. Yeah, this is really cool. I have a student now that's going to do a presentation on ocean acidification, and I'll share this video with him because I didn't know that. I mean, I know in general it's a threat to living organisms, but you were specifically saying it's a threat to the oysters. A question, another thing I didn't know was that oysters were rich in omega-3 fatty acids. Um, my cousin here is doing research in that area, omega-3 fatty acids in relation to bipolar disorder. So that's interesting. What I wanted to ask though is, I understand that you do microbial analysis of the oysters. have you guys started looking at elemental analysis for lead, arsenic, some of those things as they are
I did.
filtered? Yeah.
Yes, I did. So we did have a very good collection of what we call is the data on lead, arsenic, mercury, and cadmium. These four
Mhm.
parameters,
Yeah.
and we even have results, and that is like completely like another topic for
Right.
presentation.
Right.
It's like it is like 2 3 year studies with Oyster, like study sites, in
Mm.
our study sites. I do remember doing that study in a different estuary. I
Mm.
didn't do it where we study, but it could be a very good. I did. So here is what we did. We did use one of these what we call on-site monitoring device with what we call the, not the arsenic but mercury,
Mhm.
and I would say cadmium.
Mm.
Yes. And so what we did was we actually monitor with this instrumentation, but we never went too far that I needed to have confirmation, lab confirmation of the concentration. So I kept the samples preserved. So I am hopeful that we are going to finish that study,
Okay.
and we will be able to have some results from these sites in terms of like a cadmium, arsenic, and different parameters. But we I can't always like know exactly how on-site monitoring effect. See, all these sensor, we have a capability of calibration, but
Mhm.
we never had a chance to compare that with another methods. So
Mhm.
that's why I don't present it until I am sure of it. That
Right.
concentration is a good reflection of what we see. Lab methods generally give you a much detail,
You're right.
concentration. Yeah.
Right.
So that's but we did absolutely, and we do see it. We do see it. We did. Did you know we did microplastic research in that area?
Oh, really?
Yeah,
Okay.
we also did like microplastic uh, research. The student collected a lot of data, and we are still like analyzing some of our samples. And Dr. Ali has been helping it with us. And what we need to have the chemistry folks to join us, because we
Mhm.
need the chemistry, elemental analysis of the samples. Um, what is this plastic microplastics are coming from? Is
Mhm.
like what are they made of?
Mhm.
And how do they, basically in the system persist? So these are the things that we are looking at. Again, we got students involved in, but undergraduate, one of the biggest challenge we have is that students, they have to concentrate their academic schoolwork. And
Mhm.
sometimes we don't get to finish all the project we started,
Right.
but,
Right.
and Dr. Murray, from your
Yep.
experience.
This is
Yeah,
true. So, because that's a very important aspect because it has to do directly with like consumption, right? Of the stuff.
right,
You need to
right.
know what's in the oysters, what they're eating, and there could be guidelines developed based on that type of, research. So,
Right,
yeah.
absolutely, absolutely.
All right. Other questions, comments?
Hi, Dr. Ozbay.
Oh, hi, Omar. Thank you for joining. Thank
Yeah,
you.
thank you. Good morning. It was to all. It was a really like excellent presentation. Thank you for sharing such insightful information with us. So, Dr. Ozbay, I have one question. So, you studied like most of the microbes. Is all are bacteria, or also do you have some fungal species that you identified that oysters can clean some fungal species?
Oh, so yeah, we never did the fungal. Oh my God, this is very interesting. In fact, we did some sediment work with Mohana's project, and I think more often you could see the fungal communities associated with the sediments. And so we
Okay.
haven't finished those analysis, but it is a great question. Um, we don't get to see a lot, but fungal community also exists at the aquaculture site, if
Okay.
the oysters are in the interface between the air and the water, you get to see those kind of communities. But I didn't specifically monitor it, but I know they exist there. But it is a good question, because we do it. So like whenever we do diatom project with what we call with the on the rocks around the sediment area, we also believe that there is also fungal communities a lot of times, but we don't really expect to see them suspended too much in the water unless like they are apart. So, but that is a good question. We didn't really do specifically work on that one, but I know what you're talking about. It's like they generally exist in that communities based on my background previously. Yeah.
Yeah. Thank you, Dr. Ozbay.
Thank you. I have a personal question.
Yeah.
Do you have a favorite oyster dish?
Do I have a favorite oyster dish? Oh my God. So, Dr. Murray, I am terrible about not following up what I just say. It's like I love eating oyster roe with with tomato sauce, like a spicy tomato sauce.
Okay.
Or like literally I am a raw oyster eater, but I tell you, I love mussel dishes. Like Michelis, it is blue mussel and with garlic. With
Okay.
garlic, and it is like literally just stew it with the garlic and little bit vinegar, and it tastes so good. And but with oysters, I one time I just ate from one of these conferences. They actually breaded the oyster, what is it called,
Okay.
meat, and within their own shell, like it was just so beautiful. What we call is the presentation of food. They kept, so they breaded it. They actually fry the breaded oysters, the tissue, and then they actually put them all together in a like a little bit a container. And what they did is that they put the vegetables around it. The shells were boiled. They boiled the shells. It was very complicated. I thought it was a lot of effort. This was one of the conference.
Okay. Ellis, are you hearing this? Is it making your mouth water? Ellis, you were saying earlier that you like oyster dishes too?
Well, I don't know if he's on or not. But
Okay, here I am.
go ahead.
Okay.
Yeah,
Oh,
it's
thank
very
you.
interesting. My question is, I'm a big lover of smoked oysters,
Smoked. Okay.
and I want to know your opinion about smoked oysters.
I love smoked oysters. Oh my God. Thank you. I like smoked salmon. I like smoked oysters. And believe me, I ate like a number of them. And I came from a originally community that was in fishing town. They did smoke things a lot in my fishing town. And I remember they didn't have oyster necessarily in that particular area, but they did have mussels, blue mussels. They actually smoked that too. So I remember that. Oh, thank you for bringing this up. Yes, I like the smoked oysters. Yeah, I completely forget about it. Yes. It's,
All right, thank you.
All right, thank you. Anybody, we could probably take one or two.
I'm just gonna have a comment. It's something that the Benton Harbor city manager is now retired, and he can be on your Environmental Fridays all the time. But what I want to say, Dr. Murray, I first want to say thank you very much for the speaker. Very enlightening. Thank you. And I can see how you and Dr. Murray has so much in common, connecting young people and students in the community involved in our environment. This Environmental Fridays is such a wonderful environmental education component. And so briefly, I mean, I don't know if you guys were on the show, we've had a community food hub expert speak last week. She came from India and she talked about so many things, but also about recycling and what they're doing in India. And so with this show too, this was just wonderful. But I wanted to say, Dr. Murray, you must continue doing this, because already today, Benton Harbor Area Schools says they would like your edited versions brought into their school district. So that is saying a lot. Community Action Group out of Ohio are interested in speaking with you. And we had a pre-med student from North Carolina who was like, wow, I had to get off the call, but can you let him know that he's going to talk to his professors about what you're doing with Environmental Fridays? So 5 years ago, you couldn't have told me anything that much about the environment, maybe not even 4 years ago. And now I'm on this show, and I think there is a former commissioner from Benton Harbor, Mary Ellis Adams. And she says this all the time. Ooh, every time I leave off this show, I learn so much.
Me
so
too.
with that being said,
Me too.
and to everyone on here, thank you for exciting us about the environment and educating us. And I would have never known or even thought about being interested in some of these subjects if it wasn't for what we're doing with environmental education here. So I'll keep it short, and I'll reserve my other questions until later because we don't have a lot of time. Thanks.
Yeah, I think our speaker today was the first speaker that has this phrase, oh my God. I'm like, she is so excited about her work. Like, it's infectious. Keep it up. I could see why you have a lot of students working with you and wanting to work with you. That is really, really cool.
Thank you, Dr. Murray. I really appreciate, I look at your programs. It is so, oh my God. You are a very dedicated person. Yeah, we have a similar personality here. You are very excited about things. I am too. It is so great to be part of this. And thank you for making me.
Thank you. All right. So I think if we don't have any other comments, we wrap up.
Dr. Murray, this is Amarnatha Venupusa from Delaware State University.
Okay.
Today, like as you mentioned, there is a slot on December 6th. So I would like to give you a presentation on climate resilient crops. And
okay, so yeah, we could talk offline about it some more.
Okay, sure. Thank you.
All
Yeah,
right.
that's what I wanted to. Thank you, Dr. Ozbay, for sharing this opportunity. Yeah.
All right.
Of course. But I want to tell all the Delaware State people, I have Dr. Alvarez on the call. He's an associate dean for land grant research. And all the other students and my colleagues and scientists, please continue being part of this program with Dr. Murray. So let's consider me as a start. Please be part of this. This is a wonderful program that I expect you to support it and be there, be part of it, be present when they have great presentations. And thank you again. Thank you so much.
All right, what she said. I'm not going to repeat it. What she said. Thank you so very much to everyone, our guest speaker. Thank you for all of our audience members. Thank you, Director Shaw, for coming on and showing your support also. And if it's Friday, it is Environmental Fridays. It is personal. Talk to you guys. See you around next Friday. Take care.
Thank you very much.
Thank you.
Thank you.
Bye-bye.
Bye, everybody.
Thank
Have
you
a good
so much.
weekend.
Thank you.
Enjoyed
Bye-bye.
it. Bye-bye.
Bye-bye.
There's one before you.
Uh-huh.
This is not
Thank
about
you.
the Environmental Friday, but I observe or I notice or I know that you'll be on the USC weekend panel in a couple of weeks.
Yes. Yeah, they recruited me for that. I don't know why, but whatever. So you'll be there at that?
Yeah, I've been.
All right, should be interesting.
What is your topic? What's the topic of the?
I don't know if they've decided on a topic yet, for, a final topic. So we'll see.
Okay. All right.
All right. Take care.
Same to you. Bye-bye.
Bye-bye.